Good morning, everyone. Last class we did what? Mutations, and we focused on. And I basically introduced you what is a mutation and what are different types of mutations. And I told you this comes in the exam very often to write the types of mutations. And then I gave you one question on meaning just to tell you that the type of mutations can be asked in several ways. Meaning the next thing written in your syllabus is in the NEP syllabus is the molecular basis of mutation. So molecular basis of mutation, what we'll be studying here is that of course in point mutation one base is changed, in deletion a segment of chromosome is gone, in duplication it gets duplicated and the expression is different. But how this happens, meaning how a mutation happens, and if you remember we talked about spontaneous and induced mutations. So it would always be in interest to induce mutations. We talked about that if you want to study mutations, right, we should know how to induce them. Then only we can study, right? And if you go further, you will realize that the mutations we are talking, they cause mutation in the human body, but in chemical warfare during World War, there was a mustard gas which was used, and mustard gas did what? It caused the mutation and then the death. See, even now, very often we discuss that this uh, COVID was again a warfare. So mutants have been a very interesting and a common warfare substance. Meaning, if you want to destroy anyone, you spread a mutant there and the organism will be destroyed. So we'll be talking about spontaneous mutation and induced mutation, but our purpose of studying this is what? Is that how these mutations are induced. Meaning what happens when we expose them. First, what happens in nature. So I divided this lecture into two parts again because it becomes very lengthy and then you are not left to ask questions here. So if it is, first we do the spontaneous mutation and then we'll go to the induced mutation. So in the spontaneous mutations, if you remember, if you see the title which I had kept was why it is interesting, the why should we, what actually happens in the cell when the spontaneous mutations are being induced, what happens in the cell, what does that mean, meaning what happens in the cell naturally that the cell changes its DNA composition, right? So we are first talking about spontaneous mutation, that inside a normal cell what all keeps happening. So we'll be studying this under 5 heads. One is DNA replication and slippage. Then we have tautomeric shifts. Then we have deamination and depurination, oxidative damage and transposons. I will take all of them one by one in details. But with naming out these processes, I'm just trying to tell you that I'll be talking about 5 processes, right? One is DNA replication and slippage which happens. Then tautomerism, so apro chemistry maybe you must be studying a lot. Deamination, depurination and oxidative damage and transposons. Transposons again is written in your syllabus and it is important to study transposons. So I was just telling you that in the last class or some classes before I taught you DNA nucleotide structures, if you remember. Cytosine is what? You remember that? Uh, Cytosine is 2-oxy 4-amino.
Four amino pyrimidine. Then we have uracil. Uracil is 2,4-dioxypyrimidine. Thymidine is 2,4-dioxy 5-methyl pyrimidine. structures because we would be needing them again while studying the mechanism of mutations. Guanine is 2 amino 6 oxy purine and adenine is 6 amino purine. So we were talking how these spontaneous mutations are happening. Here is DNA replication errors and slippage. So what happens in DNA application errors and slippage is that we have mispairing very often. That the DNA polymerase, we know, it is prone to mismatch and insert a wrong base, which cannot be corrected even by 3' to 5' exonuclease proofreading capacity of this DNA polymerase, and this leads to mutation. So when a wrong base is inserted and that is not corrected, so the base which we were expecting is changed, and this is what point mutation is. So the replication errors very often are the reason for mutation, and another good reason which is why the mutations are happening is missing points and slippage. That is, that DNA strands, sometimes the double helical strands, it forms a loop if it does not have a matching base pair in front of that, and when the DNA template loops out, that portion of DNA is not replicated. So during replication it is slipped and missed, and that leads to deletions. So that region of unpaired loop, it is missed in the synthesis by DNA polymerase in the newly synthesized strand, and this leads to what? This leads to deletion which is being created. So in routine, in a normal DNA replication, in normal circumstances in a cell, the insertion and deletions happen because of DNA replication errors and slippage. And I hope we remember from the last class once again that we remember that insertions and deletions would lead to frame shift mutations, amino acid insertions and deletions in the gene product. So this is the one region which keeps happening in the cell, and another reason is tautomeric shifts, which is a process basically which is normally happening in the cell. So in tautomeric shifts, what happens is the nucleotides, they change their chemical forms. So first, what is a tautomer? Tautomer is the alternate chemical form that differ only by one proton shift in the molecule. So the nucleotides, the purine and pyrimidines, they exist in different tautomeric forms, and in different tautomeric forms they have different binding affinities. Like for example, if I talk of thymine is, as we talked just now, is 2,4-dioxypyrimidine in its keto form, but if it is by tautomerism converted to thymine enol form, in keto form it binds to adenine, that is an amino base, so it will bind to adenine normal by 2 bonds. But if by tautomerism it changes to thymine enol form, it will bind to guanine by 3 bonds. So what happens as a result of this is that we have thymine base pair changed to the thymine AT base pair is changed to TG, and G in next generation will be binding to C. So it's a substitution mutation that AT base pair is changed to GC base pair. So tautomerism is a very important phenomenon which is happening inside the cell, normal circumstances, in the normal situations. Another example, if you take the example of cytosine normally is 2-amino-4-oxypyrimidine, but it binds to guanine by, which is a keto, and it binds to guanine by 3 bonds in normal circumstances. But cytosine in the amino form, by tautomerism, by proton shift, if it converts to cytosine amino form, what it will do is now it will bind to adenine, that is amino form. So cytosine now will bind to adenine and by just two bonds. So what will happen as a result of this? Again, GC base pair is changed to a AT base pair. So tautomerism is very common and it leads to changes in the base pairs. And this base pair change or the mismatch, it leads to what? It leads to different bases coming up and occupying in the next generation. So if you see the base pairing, I've just shown the tautomeric shifts in the replication of DNA for next few generations. You see that during semi-conservative replication, it leads to shift in the base pair and it leads to transition mutations occurring. So tautomerism is another phenomenon which is very common and which leads to mutations in the normal. It's a type of spontaneous mutation, meaning it no stimulus is present from outside and it is just happening in nature in the cell. The next process which we thought we'll cover today is depurination and deamination. Now what is depurination? Depurination is the loss of one of the nitrogenous base in an intact double helical molecule. What does it mean? It means that it's a natural phenomenon in a cell that it has a tendency to lose its purines. Purines are guanine and adenine. So inside a cell, it normally happens that purines, which are bound by a glycosidic bond to the deoxyribose sugar at number 9 position, the bond is broken and it releases the purine from there and it leaves a blank site. So when the site is blank, what happens? This is a space where any base pair can come and occupy. So it leads to change in the base pair, and if this apurinic site is not repaired by the cell in the normal circumstances, and it's a space, it's a gap where any nucleotide come randomly and occupy. So it leads to maybe the purine, whatever was present, will be replaced by any of the rest of the base pairs, which leads to mutations. And now we have already studied that these mutations could be maybe changing the frame, or it may be very important, it may change the frame and lead to termination codon or anything, but this mutation might have an important impact in protein synthesis and gene expression. So that was another important phenomenon in spontaneous mutations. And one more we feel like is deamination. Deamination is what? Is that amino group in nature is converted to keto group naturally in the cell spontaneously, and sometimes as a result of certain mutagens which have come in the cell. But what happens as a result of this is that amino group is converted to keto group. And because in the beginning we learned the structures, how important it is to have an amino group and a keto group in the bonding of nucleotides also, and in maintaining the intact structure of the bases also. So if this is changed, like say for example, if we change the cytosine, if we change the amino group of this to keto group, it converts to uracil, and uracil will bind to A rather than G. And if we talk of adenine, same thing happens in adenine. Adenine by deamination converts to hypoxanthine, and hypoxanthine will not bind to T. It will bind to a different base pairs. So these small changes keeps happening in the normal cells, but they explain you the basis of induced mutations also at several places. So they are important and they explain you the basis of induced mutation, but they happen spontaneously inside the cell also. So the major effect again will be the change in the base pairing specificity, and we have different mismatches leading to the different bases coming and occupying the space, and it leads to changes or the mutations. The fourth method which we wanted to study here is oxidative damage. is that DNA in nature, by nature, cell has several processing going on in them, and during these processes what happens is several byproducts are synthesized, and one important interesting byproduct is reactive oxygen, which is produced in aerobic respiration. The reactive oxygen, like for example, the superoxide or maybe hydroxyl radicals or hydrogen peroxide. So during normal cellular metabolism, these compounds are created, and these compounds are natural and a constant threat to the integrity of DNA. So what does that mean? That they will be there in the cell and they will come and damage and change the DNA. So it leads to chemical modifications in DNA, and these modification could lead to change in the base, losses of the base, or single-stranded breaks in the DNA. And now we know about hundreds of such different changes, and I am short of time here. I cannot explain you all those changes, but what I can say to summarize is that these oxygens, the reactive oxygen species like superoxide, hydroxyl radicals, hydrogen peroxide, they are also a big reason for mutations and the spontaneous mutations which are occurring in the cell. Another interesting thing, and which is specifically written in your syllabus, is transposons. So if anyone knows what is a transposon and how do you define transposons? Yeah, they are jumping genes, and if you want to know more about them, that transposons are the elements, or they can move from one DNA to the another DNA, between the genomes or between the DNA elements. But when they go, they don't go alone. They carry some genes with them, some DNA segments with them. So they occur in bacteria, humans, everywhere. And transposons are natural mutagens. They move from one location to another, and they insert themselves into the new region, and they alter the reading frame altogether. So we have maybe a stop codon is introduced, or the reading frame is changed, and instead of one protein, now we have a different protein synthesized. And otherwise also, these transposons are known to create chromosomal damage, that is including the double-stranded breaks, inversions, translocations, and in a way transposons is a new chapter if I go and teach you there, but here I was expected to teach you in terms of mutations. So they cause big mutations, and they are very important, and they cannot be ignored, but they are a big source of spontaneous mutations in a cell. So that was it. So I was talking in this section, or maybe I thought I can, because I wanted to study this in two parts, teach you in two parts. So here I taught you spontaneous mutations, molecular basis of spontaneous mutations, and we could cover the 5 types or 5 methods or 5 mechanisms, and we'll be talking about how we can use these mechanisms in inducing mutations. But before we disperse, because I will take that in a separate class next time, I like to present in front of you an interesting question which comes out of the study. Is that, I'll just read it out for you, is one of the most famous cases of X-linked recessive mutations in humans is that of hemophilia. It is found in descendants of Britain's Queen Victoria, that we all know, that the Britain's Queen Victoria is carrying X-linked mutation for hemophilia. And the pedigree analysis of the royal family indicates that Victoria was heterozygous for that trait. However, her father was not affected. Her father did not give her this gene, and there is no evidence that her mother was a carrier. So for Queen Victoria, what I am saying is that she did not get this gene from her father, nor her mother was carrying this gene. So can you please give me some possible explanation that how the mutation arose in Queen Victoria? What type of mutations could lead to these this structure or this disease? Any guesses? Anyone wants to guess what could be the possible reason for this?
Ma'am, I think during the replication process, like cell division happens, cell cycle happens. So during that time, an error could occur. So that's why a mutation arises. It could be a point mutation, but after several replications, it has turned into a large nucleotide deletion or an addition, which could lead to this mutation. And ma'am, there are several bases of mutations like DNA replication and slippage and tautomerism. Manya. Yes. Ma'am, as I just told you, it could be due to DNA replication slippage, it could be tautomeric, and it could be depurination and deamination, oxidative damage, and transposons. Yes.